Hi there. I'm Chad Briscoe. I'm the Executive Vice President of Bioanalytical Sciences at Celerion. I oversee the bioanalytical laboratories in Lincoln, Nebraska and Zurich, Switzerland for Celerion. Today, I wanted to talk to you a little bit about how automation has changed in bioanalytical laboratories over the years, and how that's changed the way that bioanalytical laboratories serve the pharmaceutical industry. Uh, Bioanalytical labs, it's challenging to do automation because the assays are widely varied. They're typically niche assays. They're used for a relatively small number of samples, thousands to tens of thousands, really at the most. And so, any automation applications need to be highly flexible to work with many different types of assays that vary greatly. However, advances over the years in quality of the processes that we use, the precision of the instruments that are utilized in the automation, and frankly, the growing numbers of samples in different types of studies has made it so that the investment in automation really is worthwhile. And that's as it often happens with with technological advances, that's really made an impact, and that impact has been made possible because at the same time as automation is getting more accessible, the timelines required to develop assays is shortening. The timelines to analyze samples is getting shorter and shorter. The development and requirements that we have is simply probably a half to a quarter of the time in the expectations. We all know that time is money in the pharmaceutical industry. So the faster we can conduct our analysis, the faster we can develop drugs, and the sooner we can get those drugs to patients. Not too long ago, in a conversation I was having with a bioanalytical colleague, they told me that they considered Celerion one of the most automated bioanalytical labs in the industry. And so, what does that mean? Well, I like to tell a story often when people ask me about that. When I started at Celerion and went and visited our Zurich lab, we have there probably around 20 different types of robots, Hamiltons, T-cans, all sorts of different tools. And often when I go into those laboratories, I see these automated systems are sitting dusty in the corner. And that day, 18 out of the 20 instruments had been used at some point during the day to drive the processes. And and when you're using the automation to that extent, it simply is just part of the way you do the work. And nowadays in our laboratories, the lab analysts and the management and the customers we work with, they almost can't imagine doing assays that aren't automated when the capability is there to automate those assays. So one of the challenges of automating bioanalytical laboratories is each of these different assay types, be it an LC-MS/MS assay, a ligand binding assay, or flow cytometry or PCR, they all have different requirements and needs, and so that does require a diverse talent set in order to optimize the automation. But it requires a wide range of instrumentation. The needs of an LC-MS/MS assay are different from a ligand binding assay, are different from a flow cytometry assay. And so we've developed the processes over many years to optimize which instruments are the best instruments, the right instruments for each type of assay uh, in the workflow. Of course, automation isn't just about the instruments. Automation is also about the systems and the way that we collect the data, the way that we move the data through the sample and through the sample analysis process. So having the best instruments is great, but you really need to have the LIMS system. You need to have the electronic lab notebook. You have to have an efficient way to report out the data in order to efficiently take advantage of that automation. So, for instance, within Celerion, our phase one clinical pharmacology units, which have a proprietary electronic data capture system called ClinQuik. We've interfaced that directly with our automation. So studies that come from our phase 1 clinics, the data flows seamlessly into our Watson LIMS with the samples, and that data flows in and out of our electronic lab notebook and, of course, the automation platforms. Ultimately, on the back end, of course, that data is processed through the system, and then we're able to report that data with our SelectSYS portal to deliver that data almost real time to the sponsors. Another question I'm often asked is about how much does automation really impact your processes? Everybody wants to know. Can you do twice as much work? Can you do 4 times as much work? And I had the same question when I was visiting the laboratories and looking at all this amazing automation. And it's really been a journey at Celerion. So if you look back when the automation was first implemented, there were a lot of advantages that were seen, but the automation itself didn't make as much impact in the actual throughput. It was more about quality, consistency, robust assays. But as the automation has matured over the last 10 or 15 years, we've seen a 2x gain, 4x gain. Now we're seeing with some assays, we're seeing as much as a 6x increase in our throughput, and that's enabled us to run over 500,000 samples a year between our laboratories on really much smaller numbers of staff than I've seen in the past. One person can process, as I said, 4 to 6 or more, 6 times or more samples, with LCMS and ligand binding assays than we saw in the past. And again, that's done with higher quality, higher compliance, higher degree of data integrity. At Celerion, one of the processes that's highly integrated throughout is our connection between our clinical pharmacology unit and our bioanalytical laboratories. And we have many ways that our clinical pharmacology units and our bioanalytical labs collaborate. But one of the really unique aspects of how we collaborate is our co-location of our lab and clinic in Lincoln, Nebraska. So in Lincoln, our laboratories literally sit on top of our clinical pharmacology unit. And so when we have to collaborate between the sites, we can bring the clin farm people and the lab people in the same room for meetings. We can meet with the customers together. But where it really makes a difference is not just in that planning and that oversight, but it's the actual execution of the study. So there's a number of different ways that's been really beneficial for the studies that we've run. First of all, you can walk the samples right upstairs and you can put them right on the instrument. So we, whether it's LCMS or flow cytometry or ligand binding assays, we're able to bring those samples right in and run them literally same day. In the case of flow cytometry, where we have very limited stability with some assays, we're running those assays within hours. If there's no stability available or stability can't be established because of the assay type, we can get those on the instrument immediately. Another way that this adds a tremendous amount of quality to the work is that we have this seamless process from our ClinQuik electronic data capture system in the clinic through to Watson LIMS into our electronic lab notebook, and then reporting out the data through the back end. And so things like sample mix-ups, samples lost in shipping, even those 1 day to ship samples, 1 or 2 days to ship samples, is no longer an issue. And recently we've also invested in one other aspect to up the game a little bit, and that's with our PBMC isolation services. So we recently launched PBMC isolation services across all of our clinics. And so we utilize our bioanalytical laboratory cell biology experts. It's really a team of PhDs who've developed the processes and trained our lab analysts across our clinical network to process those samples that need to happen real time within hours, get them into the freezer when they need to be frozen and shipped, and then they're available for biomarker analysis in short term or even down the road. We see a lot of customers now that collect them with future analysis in mind. So at Celerion, we've got a fantastic collaborative team. Our executive team has been intact since really before Celerion was founded. While I joined the company Celerion just a little over 2 years ago, I used to work at MDS, which was Celerion's predecessor company. So really, our executive team has been together since day one of the company, and that has enabled us to have a culture of quality and excellence in everything we do. One example of that is certainly our laboratory automation that we've talked a lot about recently. But it goes across many different systems in the way that we've integrated those systems, and we've been recognized for many years straight now for our the CRO Industry Leadership Awards, and that doesn't come without a lot of hard work, a lot of collaboration, and everyone pulling in the same direction and operating on the same wavelength. Celerion is a company that has been highly invested in over the years, and that's enabled us to keep up to date in the automation and equipment and tools that we use. We're constantly investing a large percentage of the money we make back into the organization to make sure that we stay current with all our tools, whether it's the latest LCMS systems, flow cytometry, ligand binding, or automation. And two of the new tools that I'm really excited about in the laboratories are one, the Gyro Lab. So we've recently invested in the Gyro Lab Expand. It's taken off fast. We see a lot of demand for it, whether it's rapid method development, minimizing sample volumes, or simply adding that automated high quality of sample processing to the system. The other system that I'm excited about is even more flexible than that. So in our Zurich laboratory right now, we're implementing an end-to-end fully automated system where we literally are able to put the tubes or the plates with the samples in them on one end of the system. It pipettes the tubes, it gets them through the sample processing, it gets them onto the plates. The plates are picked up and they're moved over and set on the MSD in red, and that data comes out automatically. So that's really the holy grail of where we're trying to get, where you drop tubes on the system, and on the back end you just get data. Well, sometimes when I'm talking about automation or efficiencies or new technologies and tools, the question comes up to ask, well, are you going to need lab analysts, or are you going to need report writers if you automate report writing? And the answer is without a doubt 100% yes. There's always different types of tasks and things that those scientists and those technical staff need to do. There's no doubt that the goal is to limit the number of staff and limit the staff involvement, but I have a firm belief that if you can automate out the simple repetitive tasks, you're going to find higher added value tasks for those folks to do. If it's a lab analyst, let's get them doing more scientific work. These are smart folks that a lot of times we're asking them to spend a lot of time pipetting, but they can certainly get out there and help us to develop new assays, new innovations. For tomorrow, if it's technical writers where we're implementing an automated report writing tool, they can get in and they can implement and update and upgrade and work on driving quality processes. There's so many tasks to do, and we're growing rapidly at Celerion, so there's no shortage of work, and we're excited that we're able to take some of these repetitive tasks off of people's plates. One of the discussions I often have with sponsors is I get questioned, are you really telling, are you telling the truth here? Is, when you were in Zurich, were you really seeing 18, different automation tools running in the same day? Is this a real thing? Because they've gone to different labs, or maybe, in a lot of cases, they know in their own lab, they have these automation platforms that are sitting in the corner, getting dusty, not really used. And a lot of people can relate to that. So I would invite our sponsors, or I would invite people who want to see the automation to come visit us, come see it in action. We can talk about numbers all day. We can talk about what we're doing, but it's really, in the end, it's seeing it, it's experiencing it. It's the high quality consistency of the data that really means something. Today I was looking at the performance numbers for the lab for sample analysis across the board, across the primarily ligand binding and LCMS, where we can see the actual batch pass fail success rates. And over the years, those have just gone up and up and up in our labs, and are really, I'd say they're within that margin of error now, where it's really just, kind of random different errors that happen. But otherwise, the quality is so high now that you really can't achieve that without some degree of automation. Someone asked me, probably I've been asked a bunch of times, to describe what it's like to work in a bioanalytical lab versus, and I think particularly in a bioanalytical CRO versus pharma, or maybe versus a university research lab. And it can be chaotic, but it can also be exciting. So if I were to try and sum it up in one word, I probably couldn't come up with one word, but another way that I've also described it is, it's like emergency room science. So if you think about projects come in, they're not there's not a consistent flow to the business. You don't know when they're coming in. Sometimes the sample numbers are small and they're challenging, and sometimes there's huge numbers of samples and they're not as challenging. So that's where we've leveraged all sorts of different tools and solutions across Celerion, whether it's the use of automation for those very high throughput, high complexity, or highly simple systems. There's advantages that can be gained no matter how you approach it. So we're solving our emergency room science. One way is through automation.